Okay, so in this lecture, we'll be going over the pentose phosphate pathway. So let me just erase this real quick. So this is let me just zoom in on this. So this is just a general overview of all the reactions and substrates used in the pentose phosphate pathway. I think I will mention now is it's divided into two phases. So you have the oxidative phase in the non-oxidative phase. And we'll talk about when the pentose phosphate pathway would run the oxidative phase versus the non-oxidative phase. Um, I think to know about those two phases is they're not linear, and so even though they come after each other, they're two. Think of them as just two separate pathways. But yeah, even though they come after each other, they're usually not ran linearly to each other. So the pentose phosphate pathway starts with glucose 6 phosphate. So usually you would have glucose. You would make glucose 6 phosphate with hexyl kinase. So this glucose 6 phosphate can continue into glycolysis, or it can go into the pentose phosphate pathway. So the pentose phosphate pathway actually originated and evolved because this glucose 6 phosphate can't just keep going into glycolysis. What ends up happening is we have the enzyme GAPDH, which um, right after you split and you get GAP, and so this uses NAD plus. So when you keep running, if we just keep running glucose to glucose 6 phosphate and then run glycolysis, uh, the glucose doesn't end up being the limiting reagent. It actually ends up being NAD plus. So we can't just keep sending in glucose into glycolysis because then we'll just be wasting carbons. So this pathway develops so we don't waste carbons going into glycolysis because we'll like need a lot of NAD for that. So then a lot of the glucose ends up being shunted into this pathway, which is now called the pentose phosphate pathway. It does have a lot of purposes, but it originally developed so we don't waste carbon molecules. Sometimes it can also be called the pentose shunt or the hexose monophosphate shunt. And like I said, it starts with G6P and it degrades one carbon at a time. And then this explains why the glucose tends to go in the shunt. Okay, so like I said, it starts with G6P. It actually reconnects with glycolysis via two intermediates. So those are part of the non-oxidative phase. So this is GAP fructose 6 phosphate. So the non-oxidative phase can actually make GAP and fructose 6 phosphate, and those can go back and enter glycolysis. So pentose phosphate pathway occurs in the cytosol. So this is the cell. As soon as the glucose enters in the cytosol, the glucose can uh, be shunted into the pentose phosphate pathway in the cytosol, or it can be used in glycolysis to make energy. So, like I said, it has two phases: non-oxidative and oxidative. Uh, it's non-linear, and they run separate from each other. Okay, so the two phases. So the phase one is not really phase one oxidative phase, but it's the phase that occurs the most closely to glucose 6 phosphate. So glucose 6 phosphate comes here. It's converted to a bunch of things, so this is called the oxidative phase. And then we make ribulose 5 phosphate, and then the oxidative phase stops right there, and then everything down here so would be considered the non-oxidative. Okay, zoom out. So the oxidative phase makes two NADPH. We'll talk about the enzymes and substrates that make those. And we have a hexose that's originally 6 carbons, and then it's decarboxylated to pentose, which is 5 carbon. Non-oxidative phase is just a bunch of inner conversions between the 3, 4, 5, 6, 7 carbon molecules. Eventually, we'll get ribose 5-phosphate. So this has a lot of uses in the cell, which we'll talk about. And then we can also get some glycolytic intermediates that eventually end up in glycolysis. Like I said, so here we have fructose 6-phosphate, and here we have this would be GAP. This is GAP as well, and then both of these can enter. So GAP can enter here, and then fructose 6-phosphate can enter here. So sometimes glucose gets shunted in the cell because not all of it runs into glycolysis, and if we need even more glycolysis, those kind of go back, and then if glycolysis is what the cell needs to run, then the non-oxidative phase produces GAP in fructose 6-phosphate, and then those can be shunted into glycolysis to even make more energy for the cell if that's what the cell needs. Okay, so let's talk about why you would run oxidative phase versus the non-oxidative phase. So whenever you run the oxidative phase, it's because you want to make NADPH. So this is a reducing agent. So when you guys go over fatty acid synthesis, you will see that NADPH is very important for the fatty acid synthesis. So whenever the cell wants to run fatty acid synthesis, we'll need a lot of NADPH. So this happens in a lot of the fat cells, adipocytes, or just mainly adipocytes, but any cells that want to run fatty acid synthesis in general. Another thing it can be used for is glutathione. So this is in red blood cells, and I have a slide specific to that later on. So we'll discuss that in more detail. And then it can also be used in biosynthetic reactions, nucleic acids, and neurotransmitters. Okay, so non-oxidative phase makes ribose 5-phosphate down here. So this is the oxidative. Non-oxidative would make this. So the ribose 5-phosphate. So this is actually, if you remember, DNA has a deoxyribose base, and then RNA has a ribose base. So they have for example, DNA. Let's talk about so you would have so this is an example of a nucleoside. So you have adenine, and then you have the ribose sugar base. And then it's also linked to some phosphates. This is a really bad representation, but it has all these 3 groups in there. And then the adenine can be guanine, it can be the cytosine, or any of the other bases, and then same thing with RNA. So in the non-oxidative phase, we make ribose 5-phosphate, which eventually becomes the deoxyribose for DNA, and then ribose for mRNA. So whenever the cell is replicating or transcribing a lot of things, it will need to run the non-oxidative phase. So you will see this a lot in tumor cells. So tumor cells will, because they're replicating really fast, they're dividing fast, so they want to replicate their DNA as fast as they can. So they will mainly focus on the non-oxidative phase, and they will make ribose 5-phosphate. Okay. Okay, so now let's look at the first reaction. So as far as I know, you guys should know the oxidative phase enzymes and substrates. Not sure. Yeah, I think you have to know some of the structures as well. But the non-oxidative phase, I don't think you guys have to know the structures in detail, just like you need to know the oxidative phase. I think you just need to know the general overview of the non-oxidative phase. Okay, so this is glucose 6-phosphate. It's a glucose and it has a phosphate and carbon number 6. The first reaction it runs is done by an enzyme called glucose 6-phosphate dehydrogenase. This enzyme uses NADP plus and makes NADPH. and then we make this phosphoglucono delta lactone. It's kind of hard to pronounce there, but we make this. So this is our irreversible step. It's the rate limiting. It's the committed. So as soon as we run this reaction, we're committed into making we're committed into running the oxidative phase of pentose phosphate pathway. So like I said, glucose 6-phosphate can be go into any other cells, go into any other pathways. So as soon as it goes into this reaction, we're committed into making NADPH. If we also need to run the non-oxidative phase, we need to go through the oxidative phase first. So we would also need to run this reaction to get to the non-oxidative phase. But when we get to the non-oxidative phase, we'll talk about it more because it's kind of confusing because they come after each other, but they run separately. Okay, so this enzyme, it's highly specific for NADPH. So NADPH can, if we have a lot of NADPH, it can actually go back and inhibit glucose 6-phosphate dehydrogenase. And if we have a lot of NADP plus, so this can activate it. So NADPH would inhibit it, and then NADP plus would activate it. And it can competitively inhibit it. That means it binds to the active site, not the allosteric site. Okay, so now that we have our 6-phosphoglucono delta lactone, we run it through this dehydration, not dehydration, this reaction that uses water. And what this does is it breaks this ring open, and now we have 6-phosphogluconate. And this is done by the enzyme 6-phosphogluconolactonase. Yeah, it's really hard to pronounce. This reaction is reversible, but what it does is it pulls the equilibrium for the previous reaction into producing NADPH. So now that we made this, we run into one more reaction in the oxidative phase. So this is the second NADPH that we will produce. So this enzyme takes 6-phosphogluconate NADP plus, and then we make ribulose 5-phosphate, and then we make NADPH. We have an intermediate, beta keto acid intermediate. So this reaction is irreversible as well. So an easy way to remember it is when you're making NADPH, it tends to be irreversible. And then we also have so this is a decarboxylation reaction. We don't have to really know the mechanism in detail for this reaction. Just know that NADP plus gets reduced to NADPH, and then this, not only does it get oxidized, we also lose a CO2. So now we have this ribulose 5-phosphate. If we were just running the oxidative phase, we would stop right there. If we need to run the non-oxidative phase, all of this would go into the non-oxidative phase. So the ribulose 5-phosphate can continue the non-oxidative phase, like I said. So part of this reaction is that when we convert NADP plus to NADPH, we get a lot of H plus. Because this happens in the cytosol, if we run the pentose phosphate pathway a lot, we can actually start to accumulate H plus. But then something special about the cytoplasm is that it has a lot of buffers, and it tends to compensate for all the H plus that is produced. Okay, we move on. Okay, so now this is our ribulose 5-phosphate. This is what we made from the oxidative phase. So if we just want NADPH, we would stop right there, and we would need to continue. If we need to go to the non-oxidative phase, we would run two reactions. So this would be non-oxidative phase. So if we need to run the non-oxidative phase, we would convert ribulose 5-phosphate into ribose 5-phosphate. This is done by phosphopentose isomerase. And we would also need to convert it into xylulose 5-phosphate, and this is done by phosphopentose epimerase. Another So this is it's added in there. I don't think you guys need to know it. Ribulose 5-phosphate can actually be converted into ribulose 1,5-bisphosphate, and can be involved in photosynthesis. And this is done by the enzyme phosphorybulose kinase. So if photosynthesis actually uses this ribulose 5-phosphate and then converts into ribulose 1,5-bisphosphate. Okay. So this is a general overview of what the non-oxidative phase is about. So this would be our cellulose and our ribose. So those are those two right there that we made. So what the non-oxidative phase does, we take two carbon 5 molecules and we make a C3 and a C7. Then the C3 and the C7 become C6 and C4. And then C4 and C5 eventually become C6 and C3. So this would be GAP. We'll go over these in more detail. And then the C6 is the fructose 6-phosphate. And then those are the stuff that can be fed into glycolysis afterwards. So those can go into glycolysis. Okay, so the net reaction is 3 carbon 5 molecules, and we make 2 carbon 6 and then 1 carbon 3. So the enzymes that catalyze this reaction we'll see the transketolase does the first and the last one, and then transaldolase does the second one. Transketolase transfers a 2 carbon unit, and transaldolase transfers 3 carbon units. So let's look at this in more detail. So transketolase transfers 2 carbon units. 2 carbons from here are transferred to this one. We get a 3,7 and a 3 C3 and C7, and then transaldolase transfers a 3 carbon unit. So then 3 from the C7 goes to the C3. So then we'll end up with C4 and C6, and then transketolase does 2 carbon here, and then we'll end up with a C6 and a C3. Like I said, you don't need to know the substrate structures, just know the general overview. And then a bunch of these reactions just rearrange carbons. And I have the mechanism for transaldolase and transketolase in more specific. Something about transketolase is it uses TPP, while transaldolase does not use any prosthetic groups. Okay, we move on. Okay, let's look at transketolase. So transketolase takes 2 carbon 5 molecules, and then we make 3 carbon 3, and then 3 carbon unit and a 7 carbon unit. So this would be your first reaction. It's an irreversible reaction, non-oxidative phase. So as soon as we run this reaction, we're kind of committed to running the non-oxidative phase. So our cellulose, so this is the C5, so this is the carbon donor. Ribose would be your carbon acceptors. We make a C3 and a C7. Our C3 is a glyceraldehyde 3-phosphate, and our C7 is this is really hard to pronounce, acetoheptylase 7-phosphate. Like I said, you don't need to know the structures for this. Just know that you're making a C3 and a C7 for the non-oxidative phase. Before we run this reaction, and when we'll talk about the mechanism, you'll see why this is important. Ribose 5-phosphate needs to be in the right configuration. But by that I mean like D versus L. So if it's not in the right configuration, this reaction won't actually run forward. Okay, so now we made our C3 and then the 7 carbon unit, and then we convert those into 6 carbon and a 4 carbon unit. Our 6 carbon is fructose 6-phosphate, and our 4 carbon is erythrose 4-phosphate. So this reaction is reversible, unlike the other one. And our 7 carbon unit is our carbon donor in this reaction. We give 2 carbons from the acetoheptylase 7-phosphate to glyceraldehyde 3-phosphate, and then that's how we actually give three carbons, three carbons, I'm sorry, and that's why we end up with a 6-carbon unit and a 4-carbon unit on the product side. Okay, so we have transketolase again. So transketolase does the first and the last reactions. This reaction is reversible. So something to know about enzymes is the enzyme does not really determine if a reaction is reversible or not. It's usually the substrate and the products you're making, and whether and depending on if your substrates or products are higher in energy. So this reaction actually catalyzes a reversible and an irreversible reaction in the non-oxidative phase. So transketolase takes a 4-carbon unit and a 5-carbon unit. So we take erythrose for phosphate. So we made this one in the last reaction. And then we also take a xylulose from the original reaction. Geez, I can't pronounce anything today. We take this from the original reaction. So right here. So we take one of these. And then we take the erythrose that we just made by the transaldolase. And then we run it into the last reaction, and then we get one more fructose 6-phosphate, and then another gap. So these eventually can be fed into glycolysis, and then they can generate energy there. And this whole mechanism, this carbon switching that we did from xylulose and ribose, and then eventually we got fructose 6-phosphate in GAP. So this is mainly done when you have excess ribose phosphate, and we usually do that to shunt it into glycolysis. Like I said previously, ribose 5-phosphate. So when we do the non-oxidative phase, it's because we need to make a lot of DNA, or we have transcribing a lot of mRNA. So if we have excess, it can be shunted into glycolysis with those 3 separate reactions that we talked about. Okay, so let's look at the transketolase mechanism briefly. So we have TPP here. So this is the prosthetic group I talked about. So the C2 on TPP ionizes to give a carboanion. So this is what I'm talking about. So we have the C2 on TPP ionizes, and then we have this carboanion that acts as a really good nucleophile. So this attacks a ketose substrate. So whichever one of our substrates is a ketose. And then we get this addition compound. So this addition compound rearranges, and we eventually release an aldolose. So we get our first product right here, the one in the blue, an aldolose product. So whenever transketolase runs, we get one aldolose product, but then the addition compound reacts with another aldolose substrate. Because when we release one when this happens in the cell, a bunch of different addition compounds are made, so they'll release different aldolose substrates. One of them is our product, and the other one participates in the next reaction. So this aldolose substrate reacts with the addition compound, and we make an activated glyceraldehyde. And then the glyceraldehyde rearranges, and then a couple more reactions happen. We end up kicking out the TPP. The TPP acts as the leaving group, and then we end up with a ketose product. So I summarized those in quick steps. So like I said, C2 on TPP ionizes to give a carboanion, and then we attack the substrate, the ketose substrate, and then the addition compound forms, and then we release aldolase. So this is our first product released. And then activated intermediate, so this is the activated intermediate, condenses, and we make a new ketose. So we made an aldolose and a ketose when we run the transketolase reaction. Okay, transaldolase. So Trans-ketolase, this does the 2-carbon transfer, I guess. And then trans-aldolase does the 3-carbon transfer. So we have a lysine residue, so this is near the active site, and then this is our ketose substrate. The lysine residue reacts with with a carbon on that ketose substrate, and then we make a shift base. I think that's what it's called. Can't remember how it's pronounced. Those are usually really good bases. So yeah, the shift base forms between the carbonyl of a ketone and lysine, and then the shift base eventually becomes protonated. So right here, it gets protonated, and then the bond between the C3 and the C4 carbon is split, and then we release CO2. Not CO2. We release an aldolase, and then we have this shift base with the lysine and the original ketose substrate that we attacked. So, like I said, one of the aldolase becomes our product, and the other aldolase from a different shift base that released an aldolase. So this extra aldolase substrate reacts with our protonated shift base. Well, it's not protonate anymore, but this shift base intermediate, and then we end up getting we end up using water on a hydrolysis reaction and release our ketose substrate, and then the lysine residue is released again. So the specifics of this down here is unnecessary. Just know that the aldolase attacks this carbon-carbon double bond, and then eventually the lysine is kicked out when we use water to hydrolyze the aldolase, or the ketose, and then which is released as the product. Okay. So let's talk about some regulation. So like I said, the only thing I mentioned so far is that NADPH can go back and inhibit glucose 6-phosphate dehydrogenase. So let's just refresh back. Yeah, so this reaction, the first reaction in the oxidative phase, NADPH can go back and inhibit glucose 6-phosphate dehydrogenase. So this is the only allosteric thing we should know for the pentose phosphate pathway. A lot of these reactions in the pentose phosphate pathways are reversible, so the flux and the regulation is controlled by concentration of substrates and products. So, for example, if we have another reaction that's removing substrates away, it will shift that equilibrium to run that certain phase more. So, for example, for making glycolytic intermediates like fructose 6-phosphate and GAP, the two that we made in the non-oxidative phase of the pentose phosphate pathway, this would actually pull the non-oxidative phase to produce more ribose phosphate, and then the ribose phosphate can be used in other things in the cell, like nucleoside production. So what the cell did is it kind of shunted this fructose 6-phosphate and GAP into glycolysis, just so we pull the equilibrium more toward ribose 5-phosphate. So another way it's regulated is dependent on what the cell needs. So rapidly dividing cells, for example, they'll need a lot of, because they're replicating a lot, they'll need a lot of DNA replication. So they will favor ribose 5-phosphate over NADPH because they need this for to make their DNA. So this would favor non-oxidative versus oxidative. So that's one way that two phases can be regulated depending on what the cell needs. So like I said, they're connected, but they run separately. So depending on what the cell needs is what it will decide whether it wants to make ribose 5-phosphate or NADPH. Another example I have is the adipose tissue. Needs a lot of NADPH because adipose tissue stores a lot of fats, and in fatty acid synthesis, uh, two of the reactions in when we're synthesizing fat require NADPH. So whenever we're storing acetyl-CoA into fatty acids, we'll need NADPH. So this example would favor the oxidative phase over the non-oxidative phase. And then the ribulose 5-phosphate that's made from the non-oxidative phase, because remember I said the non-oxidative phase ends at ribulose 5-phosphate. This is then, instead of being converted into ribose phosphate, it's then shunted into glycolysis. Because the cell does not need to make ribose 5-phosphate. The ribulose 5-phosphate can be shunted directly into glycolysis without having to make ribose 5-phosphate and use that for nucleoside production. So be careful with ribulose and ribose. Ribose 5-phosphate is the one used to make is the one used mainly when you're running non-oxidative phase, and that's the one used for DNA replication, like rapidly dividing cells and tumors and all of that stuff. Ribulose 5-phosphate is the product of the oxidative phase, and then once the oxidative phase is done, ribulose 5-phosphate it's kind of like just chilling in the cytosol. So then it runs the few non-oxidative phases, and then that gets shunted into glycolysis. Okay, so I mentioned that NADPH can also be used to help antioxidants. So an example of an antioxidant is glutathione. Glutathione is really important in cells that are using a lot of reactive oxygen species, like red blood cells, for example. So this is the structure of glutathione down here. So this is the reduced form of glutathione. This is the oxidized form. My pen is not working anymore. Let me just connect it real quick. So we have the reduced form and oxidized form of glutathione. So like I said, glutathione can detoxify reactive oxygen species. This is really important in oxidizing environments. So in another example, if you make an incorrect disulfide bond, glutathione can fix that, because disulfide bonds involve reduction and oxidation. So let me just write this down. So this is the oxidized form of glutathione. This is the reduced form. Okay, so this is the glutathione production. It occurs on the cytosol, and it can also be regulated by cysteine levels. So cysteine, glutamate, they're both amino acids. They come together. And then we use some ATP. We make a gamma glutamyl cytosine, and then we use a glycine. We use another ATP, and then we end up with glutathione. So this is regulated by cysteine levels. If we have a lot of cysteine, we'll make a lot of glutathione. If we have low cysteine levels, we won't make as much glutathione. Okay, let's look at what glutathione does. How many more? So this is the last slide I have. This is the last slide I have. So reactive oxygen species. So reactive oxygen species form whenever you have a lot of oxidizing environments and you have a lot of oxygen in general. Red blood cells, their whole purpose is to carry oxygen around in the blood, and they do so by having a lot of hemoglobin in the red blood cells actually don't have a nucleus, and they don't have a mitochondria. They get rid of all of that stuff, and they fill their entire cell with a bunch of hemoglobin, so they can transfer a lot of oxygen in the body. So because they transfer a lot of oxygen, a lot of those oxygens ends up becoming reactive oxygen species. So reactive oxygen species they're very dangerous. They can even cause up to cancer. So we need a way for them to get reduced. We need to reduce reactive oxygen species so they don't cause like a lot of damage. So what? So the first enzyme we have is glutathione peroxidase, and what this enzyme does, it takes glutathione, and glutathione gets oxidized, and in that process, our reactive oxygen species get reduced. So we reduce the reactive oxygen species, and in the process, glutathione becomes oxidized. So why is this important to NADPH? Why do we need a lot of NADPH when we're using glutathione? Well, to go back from glutathione, from oxidized glutathione to a reduced glutathione, we need something, we need an oxidizing agent. And we need a reducing agent, I'm sorry. We use NADPH, so this is a reducing agent. And this reduces glutathione to its reduced form, the 2GSH, and then NADPH becomes NADP, and this is done by an enzyme glutathione reductase. So this is actually really similar, and then the cycle continues. You have a reduced glutathione, so this can participate in another reaction, and then we oxidize, we reduce another reactive oxygen species, and then the cycle just continues. So whenever there's a lot of reactive oxygen species, and whenever there's antioxidants such as glutathione that are participating in the reactions, we'll need a lot of NADPH. So this is it for this lecture, I guess, or review, or whatever you want to call it. I think what I will do is I might not continue to host these live, and instead just record them and upload them, because not a lot of people are watching them, but I'll still do them for the people who watch them, because they benefit out of them. So like I said, if you guys have any questions, you can just email me and reach out, and I'll be able to help you. Thank you so much for watching.